Oh, they were wrapping up. Or,
Yes.
You have the wrong screen share. Can I just redrive for a second? Okay. Oh, I did the wrong one. Oh, duh. Okay.
Hello. Hola. Aho. Aya. Hola. And Awe. My name is Locke Blanchard. I am a current undergraduate research fellow here at Tyson University, and I'll be sharing the current site acknowledgement for Tyson Research Center. Did I say Tyson University? You didn't hear that. Located in what is now known as West St. Louis County, Missouri, Tyson Research Center operates at the intersection of research and education in the ecological and environmental sciences, sustainability, and environmentalism. Before Tyson, this land was stewarded by indigenous people since time immemorial. However, land ownership was not a concept before colonization. Therefore, many peoples have called this region home across time and even at the same time for different reasons. Despite the differences in how and when they came to call St. Louis home, many tribes have claimed to this land. Although we may never know the full extent of indigenous peoples whose ancestral lands include Tyson, we will take time to acknowledge those we do know. First, the Digi descendants of the Cahokian mound builders, who was split into 5 groups: the Ka, Rafa, Omaha, Onka, and Osage, who we know as the Kansa, Papa, Omaha, Onka, and Osage or Osage, respectively. The last of whom, the Wajache, would continue to reside in this region, while the other tribes traveled further along the Missouri and Mississippi rivers. Next, the Illinois or Illinois Confederacy, notably the Kahachia, who we know as the Kaskaskia, the Kickapoo, who we know as the Kickapoo, the Dakota, Nakota, and Lakota, and the Ojeti Shakri-Mayate, who we know as the Sioux, and finally, the Yuanchi, who we know as the Missouri people. Despite the differences in cultures, languages, and ties to this land between each of these groups, they all suffered genocide by war, disease, and cultural destruction through colonization, and all were forcibly removed from this land. We make this acknowledgement because Tyson is committed to becoming an anti-racist environmental field station and to making science more diverse, equitable, accessible, and inclusive. We start with calling these tribes by the names they call themselves, not the ones forced upon them, by uplifting their voices and contributions to science, and by centering them in how we plan the future. It is therefore vital to acknowledge the ongoing history of colonization and white supremacy, which this station is built upon, to begin the work of healing, reconciliation, and justice. However, acknowledging the indigenous history of this land is just a starting point to giving witness to the lives of these people and their impact upon this region. Therefore, we strongly encourage visiting our website, tyson.wustl.edu and exploring the About tab. There you will find resources about the indigenous history of this land, coming soon, as well as Tyson's commitment to social justice. We call for the entire Tyson community to work together as we learn and grow, and hold ourselves accountable to this mission. Enrique, Wahila, Orlan, Niwe, gracias. Thank you.
All right, it's now my distinct pleasure to introduce today's seminar speaker for the Tyson Summer Seminar Series, and that is soon-to-be Dr. Kaylee Arnold. Kaylee got her bachelor's degree in biology from the University of Redlands in California. And then she completed her master's in ecology and evolutionary biology from Tulane University. She's now near completion of her PhD at the University of Georgia, where she's been working in the lab of Dr. Nicole Dockenker in the interdisciplinary disease ecology across scales training program. Her awards and honors include the Southern Regional Education Board Doctoral Scholars Dissertation Fellowship, as well as the 2021 Trailblazing Student Award by the Ecological Society of America, and the best paper presentation for Vector, Microbe, and Disease Associations at the Entomological Society of America Conference in 2020. Her recent and forthcoming papers from her dissertation work focus on the ecology of vector-borne diseases across scales, including a major focus on effects of deforestation on microbiota associated with kissing bugs, which are the primary vectors of Chagas disease. And we're going to hear more about that work today in her presentation. I'm also very excited to let you know that starting in January, Kaylee will be joining us as an LEC postdoctoral fellow, working with Dr. Solney Edelstonson here at Tyson Research Center and Dr. Danielle Lee at Southern Illinois University Edwardsville, as well as me in the biology department at Washington University in St. Louis. She's going to be working on some really exciting work. She's proposed to study plant microbe interactions with respect to urbanization in the St. Louis region, as well as a very large focus on community and K-12 educational engagement that's integrated with her research plans. So please join me in welcoming Kaylee to Tyson Research Center. We're excited that she'll be part of our community starting soon and for a longer period of time than this week. And I hope that you'll get a chance to chat with her after the presentation as well, if you haven't already today. All right, thank you, Kaylee.
Yeah.
All right, everyone. Thank you for the invitation. I'm really excited to talk about my dissertation research. I will discuss about a chapter and a half of my dissertation research, but I'm happy to chat more with other folks if you have further questions about what I've been up to that I won't speak on today. So my title is Investigating the Gut Microbial Communities of a Disease Vector Across Fragmented Landscapes. And yes, as Rachel said, so I'm currently a PhD candidate at the University of Georgia, and I work with Nicole Dockenker. So today I will first talk about disease vectors and what they are, as well as the role of symbiotic organisms. I will talk about Chagas disease system, which is what my dissertation is focused on. And then I will kind of delve into a little bit of my dissertation research. So my work is all about the microbiome. I know this is a buzzword that you hear everywhere, that you hear often. I'm sure folks know a bit about the microbiome in both public health context and perhaps ecology context. So just as a quick primer, because I will sort of mention both words, but so microbiome, and a lot of people use them interchangeably, but they are different. So microbiome is all the microorganisms and their genes in a given system. And then a microbiota is just the microorganisms. So my work, I only deal with the microbiota. So I don't look at genes at all. I just look at what's present in my system. So microbiome, so why do we care? So I focus on the gut microbiome. So there's lots of microbiomes. There's aquatic microbiomes, there's plant microbiomes, skin microbiomes, there can be atmospheric microbiomes, but I focus on the gut. So the gut microbiome is responsible for many different processes in a given system. So that can include digestion, that can include nutrient uptake, stress tolerance, reproduction, behavior, and pathogen resistance. Let me try to use that quick enough. And then so my work, so I think about disease vectors and transmission risk. And more broadly, I ask what can influence the vector's ability to transmit infectious diseases. So first, what is a disease vector? So a disease vector is any organism that can spread disease. So that can include mosquitoes, ticks. I've heard all about the ticks out here in Tyson, pratamines, fleas, and they can transmit many diseases, including malaria, Chagas disease, Lyme disease, etc. And disease vectors are responsible for at least 17% of all infectious diseases. So again, I think about what can shape vector-borne diseases. So here we can think about transmission, what shapes transmission. So you have the pathogen itself, the vector, the host, which in this context would be perhaps vertebrates or plants, and the environment. And my research, I focus on the pathogen, the vector, as well as the environment. Also, I'm a disease ecologist, so I think about the environment. So now, what role do microorganisms play in vectors specifically? So they play an important role in a lot of different processes, but I will just kind of focus real quick on just two. So first, development. So there's the seminal papers in insect physiology by Breachard Wigglesworth back in 1944, as well as Baines et al. In 1956. And so these researchers did various experiments, and they found that if you clear the kissing bugs of an important symbiont, the one that they were able to culture at the time, if you clear the symbiont from their guts, they're unable to develop to their adult stage. So they are truly like arrested development. So that is one. So that's like one paper, or I guess several papers that was looked at decades ago, thinking about symbionts and their role in vectors. And then also we can think about pathogen resistance. So Kiramotic et al. In 2011 found that mosquitoes that had greater bacterial diversity in their guts were less susceptible to experimental infection with the malaria parasite. So this kind of goes into thinking about bacterial diversity and perhaps why diversity is important in the guts of vectors. And again, they found that greater diversity meant less susceptibility or less likely to be successfully infected in a lab. So now moving on to my study system. So I spent the last 6.5 years thinking about Chagas disease. So Chagas disease is found throughout the Americas. About 8 to 15 million people are infected, although this is probably quite an undercount. So Chagas disease is transmitted by a kissing bug or triatomine. I kind of go back and forth, but they're the same insect. So a kissing bug right there on your left. And then it is caused by a protozoan parasite, Trypanosoma cruzi. So a few things to note. So kissing bugs, so they're obligate blood feeders. So they will only feed on the blood of vertebrates. And then also the parasite is found in their feces. So they will take a blood meal from a vertebrate and then they will defecate and then their feces will have infected, or the feces will be infected. And that's how an animal can pick up the parasite. And then if it's a mammal, it can turn into Chagas disease. So an uninfected kissing bug will come to an infected mammal, will take a kissing bug, and then the kissing bug itself will become infected with the parasite. And then it'll take another blood meal from another mammal, and then it'll already have infectious parasites in its system, and it will take a blood meal, and then it will defecate infected feces. And then the mammal, perhaps after you get bit, it'll scratch, or somehow the feces will get into your bloodstream, or the animal, or whatever organism you're thinking about, but the infected feces will get into your bloodstream, and then thus the mammal will become infected and infectious and may develop Chagas disease. Also to note is that many animals will just feed on kissing bugs in the wild. So most animals actually become infected just from eating an infected bug rather than actually getting bit. But both cases can happen. So just more specifically about my study system. So I work in Panama. And so there's about 150 kissing bugs that we know of that are capable of transmitting T. cruzi. And then in Panama, there are 2 species of kissing bugs that contribute to Chagas disease. However, Rhamnius pallescens, which is my focal species, is responsible for most infections in Panama. So our pallescens are found in the crowns of royal palm trees. So the photo you see here is a photo I took in Panama of the crown of a royal palm tree. So like many areas and many nations, it is a quickly changing landscape. So that can include land use change. I think a lot about deforestation with the palm trees. And so Dr. et al. And colleagues found that deforestation is associated with increased R. abundance, as well as increased T. cruzi infection rates. So deforestation has a big play in the transmission cycle in Panama. So now my overall objective for my dissertation and what I'll talk about today is to understand the associations between Chagas disease vectors, their associated gut microbial communities, trypanosome infections, that's T. cruzi infection, and land use change. And those are my two main questions I'll kind of sort of focus on today. Is land use type associated with gut microbial diversity? And is trypanosome infection associated with gut microbial diversity? And then so overall, my research will hopefully increase knowledge just on the relationships between land use change and trypanosome infection on the gut diversity of kissing bugs. And then also hopefully this research, will help to inform vector control practices that aim to just decrease the persistence of many diseases caused by insects. So study area. So I worked throughout central Panama. So I worked at 3 sites in 2 main areas. So first I worked in Capira and La Torera. And so that was around the Panama Canal. So there are more urban areas, there's more people around, there's fewer trees. And then in total, I collected 28, where we sampled at 28 palms. And then so we had our pasture palms. These were landscapes typically not too far from where people were living. So you can just think about a pasture, usually have like 1 to 2 palms in a large area. And then we also sampled at peridomestic palms. And so these were classified as palms that were within about 50 or so feet away from any type of like residential commercial building. And then finally, we sampled at Santa Fe Veraguas, and I'll just refer to it as Veraguas from now. And then and this area was more forested, had fewer people, it had a higher density of palm trees, and then also it was higher elevation and greater rainfall in this area. So here we sampled 14 palms, and so we had our pasture palms, our peridomestic palms, and we had some forest remnants. So this area was a little bit different as we also sampled in in forest remnant areas. So for my fieldwork, this is the cool part. So I did the bulk of my fieldwork in 2017, which that's when I was a little baby PhD student. And then so the way that we collect our kissing bugs, so we get the TN of a PVC pipe, and then we put double stick tape around it. And then we place lab mice inside the PVC pipe and then kind of enclose it with these little grates. We also add some food and some like wet cotton balls for the mice. It's pretty hot on the palms. And so as you remember, or if you don't remember, but kissing bugs are obligate blood feeders. So the mouse is important to attract the bugs. And then so, as they're only found in palm trees, or mainly found in palm trees, so we would go out and then use a pole. I was never very good at it. And then try to balance the trap and put it in the center of the palm tree. And then we would leave it up there for usually up to about 3 days. And then we'd come back and retrieve them. There was once or twice where we just lost the trap and we weren't able to get it back. And then if we got lucky, the kissing bugs would get stuck on the tape, and then we'd be able to collect them, and then we would have our kissing bug samples. So just some further methods. So in total, I collected 288 samples of R+, and then we put them in 95% ethanol for storage. Each individual, and this was with the help of a lot of wonderful undergrad interns, each individual had its mid and hind guts dissected. And then we extracted DNA from the tissues. And then we also did 2 different types of sequencing. So 16S RNA sequencing, that's how we would identify the bacteria taxa. And then we also did 12S sequencing. So that's how we were able to identify blood meal species. So that's like the different types of vertebrates the bugs fed on. And they fed on, or at least we were able to identify anywhere from 1 to 5 different animals down to genus level in each bug. Most of them had like 1 or 2, but we had several that had up to 5 different species of vertebrates. And then each sample was screened for T. cruzi. So just some T. cruzi infection results. So in total, 41% of my samples were infected with T. cruzi. And then per habitat type, at the pasture palms across all areas, 30% were infected. At the paradomestic palms, 46 were infected. And then at the forest palms, 53.5 were infected. Wait, that's not right. Sorry. Yeah, we had 50% of them infected. And then so my first question was, is land use type associated with gut microbial diversity? So just going back to our study areas, we had Capira, La Torera, and Veraguas. So in our the 2 samples around the Panama Canal, and then our 1 sample at Veraguas. And so just before I get into my own results, so this was from a paper that I collaborated with a previous grad student who's now working at the CDC. And so here we looked across a similar sampling sites. So Minas and Bahamas, which I circled at the bottom, those ones are also around the Panama Canal, just different areas. And so for this study, we were looking at just the whole body bacteria of the kissing bugs. We didn't dissect them, we just kind of crushed them. But here, we're just finding a trend of that at Veraguas. So all these blue sites, and that was the site away from the canal. But the bugs collected there had greater bacterial richness or diversity that we were finding. So just based off of the old previous study, I kind of assumed that we'd have some similar patterns happening. But of course, I'm looking at the gut bacteria. So I was curious to see exactly how that may compare. So first, so this photo, you'll see this in any microbiome paper you look at. But so this is just looking at the overall composition of the bacteria found in the guts. So the biggest takeaway is that, so this is at Capira, the first panel, and then La Torera, both of these at the canal, and then Veraguas. And then so you can see just across all 3 is that the taxa that's most represented is Sphingomonas. But the biggest takeaway from this figure is that we're seeing some similar patterns from these 2 sites. However, we're seeing quite a bit of a difference just in the relative abundance of the taxa at Veraguas. And then so this figure, so this is alpha diversity. So we're just thinking about within sample diversity at each of the sites. So here at Capira and La Torera, we're showing that individuals caught at these 2 sites are showing no difference in their own individual overall diversity. But however, at Veraguas, we're showing that there's greater diversity of the bugs collected here. And so now this is beta diversity. So this is looking at, this is between samples. So this is thinking about the overall composition of each bug and then comparing it across different groups. So here we're looking at across regions. So this is really just sort of showing the same type of pattern, but thinking about bacteria composition in a different way. So here we're showing is that, again, our Capira and La Torera are showing no difference in their overall composition of the makeup of the gut bacteria. However, Veraguas, all the blue dots right there, is showing a difference. So we're seeing some dissimilar patterns between the canal sites and our Veraguas sites, or sorry, Veraguas samples. And then so now kind of going back to this relative abundance figure with the taxa. So because we're seeing differences between the big regions of our canal sites and our Veraguas sites, so here I'm going to just split them up and look at them a little more finely. So this first is just at the canal sites. I grouped them together. And then here, I lost my title, but here, this next one is at Veraguas. So we have our canal sites and our Veraguas sites. And so I only made this figure yesterday. So this is all kind of like new information that I hadn't looked too closely at. But the biggest takeaway from this figure is that, so we're seeing kind of course, different patterns of our pasture and paradomestic at canal and our forest pasture and paradomestic at the Veraguas. But all the 4 arrows, so these 4 bacteria taxa are found in canal, but not found in Veraguas. And then similarly, the 4 arrows over here, these are found at Veraguas, at least in the top 10 abundance, but not found at the canal site. So this is like actually really exciting. So I haven't gotten into function or what the bacteria are doing in the bugs. However, this just will take me to further investigation and try to understand of like, okay, some bacteria are found in one area, or at least in high abundance in one area versus a different area, and looking to see what they contribute in the physiology of the insect, and if it's important that they're lost or enriched in one area versus another. So now we're going to go back to alpha diversity. So here we have our canal sites again, and then so this is just looking at our canal sites. So we're seeing that pasture and paradomestic sites are not different, and then also at the canal sites. So so we're saying that once again, when you split apart the samples between regions, is that our forest sites are showing the greatest diversity. So we already knew that there was greater diversity at our Veragua sites, but now we're seeing that it's represented at the forest samples, and that here again, our pasture and paradomestic samples are not showing much difference in the overall bacteria diversity in the buds. So again, bringing it back to this beta diversity, so thinking about the overall composition of the bacteria within the bugs. And so this again is just at the canal site, and we're seeing no difference between our pasture and paradomestic samples. However, when we go to Veraguas and we have our forest remnants, we are seeing the forest remnants that are showing significant difference or dissimilarity in their overall composition compared to the pasture and paradomestic sites. So the biggest kind of takeaway from all these sides is that Veraguas is showing differences, and then within Veraguas, the forest sites, the forest samples are showing difference compared to the other habitat types. So now we've talked a lot about habitat type, but now let's get to kind of the bulk of it all. So infection. So is trypanosome infection associated with gut microbial diversity? So just real fast, I'll just show some previous research by some other authors. So Diaz et al. In 2016 and Arentes et al. In 2018. So these are both experimental papers. They did experimental work, and they both independently found that in a lab setting, at least, that kissing bugs that were uninfected with the parasite showed lower overall gut bacteria diversity than insects, the kissing bugs that were challenged with the parasite. So this is showing that infection is at least doing something to the gut bacteria, when just kind of independently they're showing that infected bugs show greater bacterial diversity. So now we're going back to alpha diversity, thinking about the overall bacteria diversity of each bug together. So this is just looking at infection irrespective of location. So here we're seeing that infected bugs do have greater bacterial diversity. And this is what was reflected in the experimental results. So that's always good when your experimental results reflect field results. I'm like, okay, all right. And then now, so this first panel is at canal sites, so it's splitting them up. So here, we're not quite seeing a difference between infected and uninfected samples. However, when we're just looking at Veragua samples, we are seeing a difference in infected sites, in the infected bugs. So this kind of just leads me to think a little bit about what's happening at Veraguas. And then so back to this beta diversity figure, so overall composition. So here we are seeing differences in infection status. So this is at the canal sites. And we're also seeing differences at Veragua sites. So it is a little bit different. We're at both sites, we are seeing some differences in the overall composition of infection. So we can think that infection has some type of relationship with the gut bacteria. And then however, I need to look at this a lot more, but so kind of thinking about, so first we have at the different regions, the canal sites and Veraguas, and then we teased those apart and then looked at habitat type. But so now, finding forested sites, having differences in bacterial diversity and composition, and then also at Veragua specifically. and now just pulling that apart for infection. So we're actually not really seeing a difference. I just got a p-value of like 0.09. So just sort of more to kind of like think about and analyze. However, my sample sizes drastically drop. So just kind of thinking about it, like how true is this number and what can I really conclude from this when I only have like 6 and 22 samples. But this is just something to sort of kind of ponder upon that I'm still kind of working through. But so we're seeing some patterns at forested sites, but however, we're not seeing the difference in infection right here. So just an overview of all of my results. Oh, I'm kind of talking a little bit fast. So we are finding some regional differences. So Veragua samples were significantly different than the samples collected at the two canal sites. And then also Veragua samples had a more similar community composition than the canal samples. So again, there's something happening at Veragua sites. And then also there are differences across habitat type. So 4 samples, which again, were only at Veraguas, but the 4 samples did show the greatest alpha diversity. And the 4 samples had a more similar community composition than the other habitat types. So again, so the 4 samples, which are only at Veraguas. So there's something ecologically happening there in terms of some patterns that we're seeing. And then also that there's some differences across infection status. That T. cruzi infection exhibited greater alpha diversity compared to our uninfected samples, irrespective of location. And then also that our negative samples had a more similar community composition than the infected samples. However, the infected samples at Veraguas in the forest remnants were not significantly different from the uninfected samples. So just another overview. So we're seeing differences across region and habitat type. We are seeing greater diversity at forested sites, and that T. cruzi seems to be associated with an increase in microbial diversity. So there is more to the story. So I won't get too in the weeds of this, although I did end up talking faster than I anticipated. But so I mentioned at the very beginning that we also looked at blood meal. So this is something that I haven't fully delved into, but this is sort of my current project at the moment as I'm sort of writing and analyzing. But so I have at the genus level different vertebrates that the insects fed on. So this is just looking across the region, and you can see just based off of colors, but that we are seeing some differences in the relative abundance of the blood meals they fed on. And then same thing here, just thinking about habitat type. So the bugs are feeding on different animals at different areas. And so kind of my next step is to look at those patterns and those associations and seeing if there's anything driving, or if the blood meal perhaps is driving some of these patterns. And this is a particularly important, just thinking about transmission of Chagas. So these are all, I guess, a variety of wild and domestic animals. But in the kind of more urban, more human, densely populated areas, you're only really finding your more urban dwelling animals, so more rodents. I think you'll see some sloths around the city, but there's fewer, so there's a lower richness in general that you're finding. However, when you're out in the forest, you get lots of birds. You get there's a few geckos that were found in the palm trees, and you're just finding just more animals in general. Thinking about transmission and deforestation, if you have fewer palm trees and where the animals as well as the bugs going, and just their opportunity to feed on different vertebrates. So, and I can't remember if I mentioned this, but only mammals are competent hosts. So only mammals are able to pick up the parasite and contribute to transmission. So birds and herps are not a part of the transmission cycle. So if you have more birds and herps, then there's going to be less Chagas disease transmission because they're feeding on wider diversity of vertebrates. So this is kind of like my next frontier as I'm finishing up. It's like, all right, so now how does blood meal play into the patterns that we are seeing? And kind of maybe like a little bit, what does that mean when we're thinking about habitat type and transmission? So just some further direction. So I just need to sort of further investigate the drivers of bacterial diversity. So there's a lot happening, and that's just ecology, ecological data. But I just want to further investigate what's really driving some differences, and really kind of thinking a lot about the ecology of the different areas. And then also, so measuring differential abundance, and really all this means is focusing in on the different bacterial taxa that are either more or less present in different areas. And there's not a ton of, I guess, research on the function of specific microbes in kissing bugs, especially ones that are not found in a lot of different insects, but to look further, and that perhaps maybe it is contributing more or less to pathogen resistance, really just kind of keying in on taxa that perhaps folks that are more clinical can look at a little bit more, or folks that are working on vector control can look at a little bit more. And then finally, just thinking about, so we have some, so compositional gut microbial diversity, is that actually associated with functional diversity? That's the question I ask myself every day. It's like, well, we're having some greater and lower diversity, but does it actually mean much in the terms of insect physiology and pathogen resistance? Maybe there's some microbes that have multiple functions that one, another microbe might only do one function. So thinking about how important this diversity is in the context of actually like pathogen resistance and vector control. And with that, that is all I have. So I would like to thank my entire PhD committee. They've been amazing and just kind of getting through these last 6.5 years. And then also my lab, especially Christina Varian. She is the main reason why I was able to get any bugs collected in Panama. She was on a Fulbright when I just happened to join the lab, and it was perfect timing because she could show me the ropes in Panama. And then also our amazing colleagues in Panama. I love and have been very fortunate to work with these amazing Panamanian scientists and work at both at Smithsonian and the Gorgas Memorial Institute for Health Sciences, which is kind of like maybe a loose equivalency to the CDC of what they have in Panama, and just helping me and understanding the system a lot better. And then of course my funding sources. And then last but not least, I would love to just thank everyone here for listening to me talk about my dissertation research, and also for the invitation today. And there you go.
Right, so we'll see if we get any additional questions from the YouTube channel. But in the meantime, I'll kick us off with a first question. And it was really exciting to see the blood meal data at the end and the really striking differences in which hosts seem to be fed upon and which sites. Have you thought about using the blood meal data to try to infer anything about maybe dilution effect probabilities? So the dilution effect being this idea that with greater host diversity, you can get less disease transmission if a lot of those hosts are not very good hosts for the disease. Like, yeah, if you could.
So I haven't, so it's a great question. So I haven't yet really delved into that, but that is truly is the next question of like, okay, yeah, how is it all playing into the system and kind of like what we can larger infer. But no, I haven't done anything specific, but yeah, but truly it was like, it's like, okay, blood meal data is like the last thing I need to like really focus on. But yeah, no, I think that's a great kind of like context to think about it in.
You were tracking the kissing bugs. Did you attach the PVC trap to the mouse box, and did you put the mouse box in the
Oh, so we had an individual mouse. So the box was just our like carrying box. So we would put a single mouse inside the actual PVC itself. Yeah, so we had I forget if Christina made the traps herself, but basically like the grates were like, we could open it and then attach the grate so that there was no opening for the mouse. And then you just have a single mouse in this little like PVC T end of it. Does that answer your question? Okay.
I gotta ask, were you ever working with bugs which like feed on blood? Were you ever bitten by one while working with them?
Surprisingly, I wasn't. I was fed on a lot of other bugs. Yeah, there was one day I just forgot to roll my sleeves down and then just got ate up by a bunch of like sand flies, which also carry diseases. But surprisingly, I never got hit, to my knowledge, bitten by a kissing bug.
Actually, I'm gonna read a question from YouTube real quick, because I think it's a really great one, from Derek McFarlane, who says, such a cool presentation and exciting work. Does infection persist through each of the kissing bug instars, and can it be passed from mother to egg? So as the kissing bugs are developing along their life cycle, do they keep the T. cruzi, or do they?
Yeah, so through the stages, they'll keep the parasite, but to my knowledge, it won't pass from mother to offspring.
So they have to pick it up.
Right, so the first nymphs, the babies will always have to pick it up, but then they'll carry it through. Okay. Yes, in the back. Yeah.
Do the bugs come out of the palms to eat, or do they only feed on vertebrates that are up there?
So they mostly just stay in the palm crowns, but that also really depends on how many hosts are there. So they are capable of flying quite a bit, kind of like palm to palm. But I think like if they had a choice, they would stay in the palm. Mm-hmm. Yes.
Great talk. So cool. I have two questions that are kind of related. So I noticed that in your stacked bar plots, there was about 10 genera, 10 taxa. Is that a common number to find in insect microbiomes? And then my second question is, you specifically looked at bacteria, but do fungus or other non-bacterial microbes like?
Yeah, so the first question, it's pretty common. It's pretty highly dependent on like where you're sampling, but the other researchers who've sampled in the same areas have found pretty similar like vertebrate like richness and the different hosts there. And then let's see. So yes, there's definitely other microbes in there. I don't know anything about it, but there is a particular virus that you commonly find in the kissing bugs that I don't think causes any type of like pathology, but it's just like a virus that you find in the bugs. And then there's definitely fungi. Yeah, I would just like, if anything of like me kind of sticking to like one type, also funding, and I'm like, I can do bacteria, and then one day I'll look at something else. Yeah, thank you. I guess.
Yeah, thank you so much for the talk. It's really interesting. I was just wondering if like the patterns that you observed with the deforestation and bacterial cultivation, I think that's like, have you seen other kind of have similar findings?
Yeah, so there's more studies on vertebrates, but I've been and they're seeing some similar patterns with thinking about like deforestation and like diversity of diet, so similar patterns. With vectors, I haven't quite seen I guess like good studies looking at like the deforestation in the gut bacteria. And I think, well, one thing in particular is especially thinking about different types of vectors, like mosquitoes, molting and just development, they change quite a bit from like the larval stage to the adult stage. So those patterns, so they just like kind of like re-get or, kind of reacquire a lot of their gut bacteria. But yeah, I've been looking of like, okay, and I haven't quite seen patterns, and I think a lot of it is that people haven't been quite studying it in that same way that I am. But people have been looking at like diet diversity and gut bacteria, which I think kind of like relates a little bit. Yes.
Do you know why they're called
Yeah, no, it's yeah, it's a good question. So they typically will feed on like the thin, like I guess like part of your skin. So they'll usually feed like around your mouth. So that's how they got the name. Yeah. So like if you're gonna get bit, most likely it'll be somewhere like on your face, and so a lot of times it'll be near your mouth. I guess.
Great thought.
Thank
Really
you.
good. I have a question for you. I think the blood meal analysis is so cool, and I'm a little jealous when it's so difficult. But I'm curious about the relationship between microbiome and blood meal analysis. Is anything known about feedback? Maybe that go both ways, right? Like, would the microbiome also influence both preferences for blood meals?
Yeah, I haven't seen research on that in the kissing bugs, but I think that is really interesting. And I would love to, I don't know, maybe talk to some like experimental folks and see if they've done, but I haven't seen anything in the literature myself, but I would be really curious to see if there's host preferences with the gut bacteria. I guess.
So do you look at different life stage of the PCR?
Oh, yeah. So I didn't talk about it at all. But yeah, so they have 5 nymphal stages and then an adult stage. So there's 6 total. So I have a mix of all of them. That's like a whole nother piece that I'm also trying to figure out how to navigate in my work. But also depending on the stage, they have just a different diversity of gut bacteria. So there's been some kind of competing results, but in my work, remember the adults have the greatest gut bacteria, and then the nymphs have the lowest. So even just like with all my data, it's a little bit like, well, I didn't tell you what stage they are and how much that's also influencing what bacteria you are seeing. Yeah, so it's a good question, and that's like another piece of the puzzle. Yeah.
So, like, do they have to take the lumbar or each transformation to be tested?
So they don't have to. They like they'll molt out their blood meal, but I don't think they have to as much as like ticks. But yeah, I think it's like pretty similar. Yes.
So you said you spent about a year studying Penczykowski. Did you like live in Panama for a year, or did you like travel there ever so often? I'm just curious about that.
Yeah, so I actually only spent a few weeks in Panama. So I went twice. I had all these glorious plans to go back in 2020 and do some experiments. But yeah, so but I the first time I went, and that's when I got all my field data, I stayed at the Smithsonian housing there. So I was with a bunch of like Smithsonian, mostly because I was there over the summer, mostly like summer undergrads and interns. But then when I went again to just work with the University of Panama, I just booked a hotel and like stayed in the city. Yeah. Yes.
How do you focus into did you like originally start like, hey, I want to go somewhere cool like Panama, or did you just have an interest in them, or like how did you specifically?
Yeah, that's a good question. Largely because of my advisor that I've been working with. So before, and I'm happy to chat with you later. I've studied lots of different things. In college, I studied like mammals, marine mammals. But so I got interested in gut microbiomes and infectious diseases of wildlife. And then my master's advisor, she happened to just be friends with my current advisor. They both worked in Panama for their own research. So she was like, oh, you like disease ecology, you're looking for a PhD program. So she introduced me to my advisor, and she just already worked on kissing bugs. So it was really like, oh, this is a system that's already established in her lab, and she's really passionate about it. Insects are a lot easier than vertebrates to work with when you want to do gut microbiome stuff. Fewer like permits. And then also too, and just the system itself. So I'm more excited about questions than the actual system and taxa that I study. But she was already looking at disturbance and landscape fragmentation and deforestation on the insects. And that's something that I was like, okay, how are humans having an impact on transmission of diseases? And she's like, do you want to work on chagas? I was like, all right. So that's my long-winded story of telling you that she just happened to already be working on it. And then I was like, this fits my questions. But then yes, starting January, I'll be working with like weedy plants and fungi. So yeah, I'll switch and I'll probably switch again. Yes.
So I have another question about the role of deforestation, and if you could give us any more detail about that, what that involves. So you mentioned that the kissing bugs are mostly in palm canopies, and so is it deforestation per se, or just like the fraction of trees that are palms that's critical? And are there other types of trees that could be really abundant, but not good homes for kissing bugs? I've never been to Panama, so maybe is it all palms? No,
yeah. So I guess I don't have a really good answer about like how many like non-palms are around, but to my knowledge is that you will really only find them in the palm trees. So without the palm trees, and they have the capacity to go into like dwellings. It's not their favorite spot, but really just them looking for hosts. So I guess just thinking about like just elimination of their habitat. And then I guess they'll find the palms they can find or find the host they can find. And then so the big play is the host they're feeding on. And something that my advisor found was that the type of mammal, also they're seeing some like differences in how competent even like within mammal species. So I think they found like sloths and I think a lot of rodents, some of your kind of like smaller, quicker lived mammals, but the parasite probably replicates quicker and then can, kind of the transmission cycle goes quicker. So in areas where you're only finding a couple of those type of mammals, then you're seeing more T. cruzi, and then just like also more of the bugs because they're able to reproduce quicker versus when they're in areas that are less fragmented. So that's something that she thinks a lot about in terms of like, what are those like coupling effects with the bug and what they're feeding on.
Thank you very much for the talk today. So I have two questions, more related to the palms. So one is there really only one species of palm or is it multiple species? And then two would be, what makes the palms suitable homes for them? Is it something abiotic about the environment the palm creates, or is it something biotic about the chemicals that they are able to live in, or is it sort of a combination?
Yeah, so that's a good question. So to my knowledge, just talking with the folks that are like doing more of the vector ecology, they're only find them in that one species of Adelaide Buteraceae, that specific, and also I think that's the main palm tree in Panama. And then let's see, also, what was your second question? So I think a lot of it is organic matter and then of the palm trees. And so it's so hot in Panama, but then in the palm crowns, just the microhabitat. So really I should talk about microhabitat more than anything else, but of, just like cooler temperatures. And I think they just can thrive more in that crown itself that I would believe that maybe other trees have less of, or just some differences there, but they thrive in those specific types of palms.
All right. I think that might be the last of our questions. So let's give Kaylee another big round of applause. And then I don't know what the directions are.